So today I am delighted to welcome, well, first of all, let me introduce myself. I'm Tony Gordon. I'm the VP of Genomics Business Development for Cooper Surgical, and I'm going to be your host for today. And we have a couple of excellent invited speakers. First of all, I'd like to introduce Balsam Al-Hashimi. Balsam is a registered clinical scientist, HCPC, Health and Care Professionals Council, a certified senior embryologist by CFAS, which is the Canadian Fertilities and Andrology Society. She's got a master's degree in clinical embryology from the University of Leeds, and she's currently a senior embryologist working at the London Women's Clinic, LWC, in London, UK, or London, Canada. Balsam is the lead for pre-implantation genetic testing program at LWC, a senior clinical biopsy practitioner, and an officer trainer for the STP scientist training program with the Association of Clinical Embryologists. So welcome, Balsam, and we're really looking forward to hearing your expertise in this area. The other invited external expert is Christian Ottolini, Dr. Christian Ottolini. He's the lab director at the Evewell IVF clinic on Harley Street. Christian, I've actually worked with in a research context and published, and Christian has some great research interests in the areas of aneuploidy in human embryos, which is the leading cause of pregnancy loss and other infertility issues. Interestingly, for this particular space and this particular conversation, Christian has a very personal interest in IVF, and it's not the normal personal interest in IVF that we have when people have IVF children. Christian is actually an IVF baby, one of the very first in Australia and one of the early ones in the world. So a little bit of personal connection to this area. So welcome, Christian, and really looking forward to hearing from you. Our other invited expert is from Cooper Surgical, and this is Colleen Lynch. Colleen has been working in embryology since 2004. She's got an undergraduate degree in genetics and postgraduate degree in medical genetics. She's currently working towards her PhD under Professor Darren Griffin at the University of Kent. I also have the pleasure of being Colleen's co-supervisor, and Colleen's Viva is coming up very soon, and we wish her well for that. She's worked as a clinical embryologist and quite unusually also as a genetics lab manager in a PGT lab, so combining the two together, and she's worked at Cooper Genomics since 2017. She also regularly lectures for Cooper Genomics and is an honorary lecturer at Manchester Metropolitan University on the Clinical Science MSc. Finally, I'd like to welcome my colleague, Dr. David Grimes. David, I've known for more than 15 years. He's got a PhD in molecular genetics from the University of Aberystwyth. He's worked in the field of PGT for the past 15 years, first at Bluenome and then Illumina, before joining Cooper Genomics in 2016. And David's particular expertise is the molecular biology of PGT. And I'll now hand over to David, who's going to give us an introduction to PN checking. David.
Hi, thanks, Tony. And it's great to be able to speak today. So hopefully, so this talk won't take up too much of the webinar. It's here as the introduction to really get the discussion going. But really, we're looking today at whether an embryo is 2PN or not, and whether we can test that by genetics using today's latest technology to really help answer that question ahead of any embryo transfers. So there's four main topics for today's discussion, just where we are today, and then how we can consider molecular PN checking embryos. There have been some studies looking at outcome data for the efficacy of PN checking of embryos, and how it plays into live birth. And then we'll wrap it up before we enter the guests answering their thoughts around this. There are 3 types of PGT today. So a lot of what we've discussed in this webinar series to date has been pretty much around PGTA. But of course, there are others, the PGTSR and PGTM, the structural rearrangements in monogenetic disease. Now, all of the embryos generated through IVF, no matter what you're testing them for, be it PGTA, SR or M, all undergo at the initial stages of IVF the same checks for fertilisation, the same checks around have they formed blastocysts properly. You know, that is independent of the ultimate testing that you're going to do on these embryos. And so this is something that is not just a PGTA test potentially, it could be applied to all. So today, fertilisation checking, it's deemed to be successful if there's a clear and visible 2PN. So we have the 2 polar bodies visible, 2PN during the fertilisation with 2 polar bodies visible at the edge of the oocyte. Now, typically this has been checked at 16 to 18 hours post-insemination, but again, people may vary that depending on their own experience or their own lab feelings. Equally, they may end up having to do multiple checks if it's not clear at the window that they have chosen to go and look for that fertilisation. But the reason that we check is because of the adverse outcomes that can be prevented. So we know that if a true haploid or a true triploid embryo is transferred, then we can get unwanted results such as a late miscarriage or a molar pregnancy, and both of these are an avoidable event. And so the guidelines were written that basically states that if it's a 3PN or greater, they shouldn't be transferred, and it's not recommended to transfer 1PN or 0PN. Now, is that still good advice with today's technology? So as Tony said, I am not an embryologist, I'm a molecular biologist, so this is really here as much from my remembering as anybody else's. But basically, you know, we're carrying out a fertilization check. In this case, the oocyte has been done by ICSI, but it doesn't necessarily have to have been for PGT, but this is what we've shown to show. But we have classic well-fertilized oocyte, which is nice and spherical. It's got the 2 polar bonds being extruded, the 2 pronuclei. It's all looking very, very nice, and you're very confident that this is an oocyte that is fertilized and everything is looking good. But it's not always that easy to do. So it's been well publicized and acknowledged that there are challenges in PN checking, hence the fact some people check multiple times. But some of the key things which have been mentioned is they may be faint. So are they there or are they not? They can appear or fade asynchronously, so they don't appear and fade at the same time. We have these micro PNs may be apparent. Are they a true PN or is it just a micro PN? It's hard to tell. But also, this is a visual check, looking down a microscope. So sometimes these PNs can be stacked on top of each other, which makes it very difficult to identify if it's a 2PN or not. So with all that in mind, you know, there have been various studies and we've spoken to various people, and is there a perfect time to check? Some people will give you slightly different answers, but a way around that has been to do multiple checks, checking at multiple time points just to make sure that you capture the moment. That, of course, carries a risk each time, depending on the incubator used, but you could be taking those oocytes in and out of a temperature controlled environment to do these checks, which we are trying to avoid, of course. But we know and everybody involved in IVF knows that you have embryos where you have questions around the fertilisation. Did they fertilise or did they not? How sure are we? And what do you do with those embryos? Do you grow them on to make blastocysts and then transfer on the basis of that? Do you push them to one side? You know, how do we manage these embryos? But even us from a testing laboratory, we get biopsies all the time, and we presume that they've gone through a PN check, and they're presumed in the vast majority of cases to be 2PN. But we know that around 3% of the biopsies that we test are actually triploid. So we know that even from a 2PN, scored as 2PN, some of them are triploid. So what about the other way? Which ones which are scored perhaps as 0 or 1PN or even 3PN, which are in fact 2PN? So this is really where we are. So how can we be more certain? How can molecular PN checking actually work for us in this situation? Well, DNA technology and our knowledge of DNA has advanced tremendously. Well, first genome sequence in 2001. Here we are 20 years later, and you can do a whole genome sequence routinely. We know so much about them, and what that has opened up is a huge amount of knowledge. And one of the things that we now understand is how much variation there are between individuals. And one such variation are single nucleotide polymorphisms, and that is where the polymorphisms, where one of the building blocks is different within structured DNA. So a human genome is approximately 3 billion nucleotides, or 3 billion individual building blocks. Scattered amongst those 3 million building blocks, there are these variances between people, and we can track this, and they're natural. They're nothing to be scared about in those cases. They result from natural variation, and they're part of human evolutionary reproduction. And what we have here is, if we look at the top graphic, we have, we'll call this parent A, or the mum. We have a T and an A configuration for this particular nucleotide base pair, and in parent 2, or the dad, we have G and C. So when we sequence, we can see these differences in sequence coming through from the 2 copies, one from mum and one from dad, and we can then be certain whether that DNA has come from the mum or the dad. So in a bit more detail, when I said they're scattered through the genome, that is absolutely true. And then all of us have a unique pattern of these SNPs in their genome. Now, as I said in the last slide, some of them are de novo. They are spontaneously created during the creation of the oocytes, but we inherit the vast majority. It's estimated that 99.8% of SNPs are inherited directly from parents. So it's been used for many, many years to assess the unique parental allelic contributions. And one of the tests that we run at Cooper and others have been doing for several years is basically doing this for PGTM. So rather than looking at the direct mutation, you do what's called haploblocking, and you're tracking inheritance patterns to work out the inheritance from the mum or the dad into the embryos. So in this case, we have an embryo that's come up as haploid. And we know it's haploid because it only has alleles or SNPs from the mum. And it only has one copy. So we are confident that this is now a haploid embryo. You can get different forms of triploidy. In this case, it's got 2 copies of the father, but it's a triploid embryo, and we can track, because we have mum and dad's DNA, the SNPs from mum and 2 copies from dad. So in this case, it would be a male triploid embryo. So we can really accurately track this information into the embryos. Now, as I have just stated, we do need mum and dad's DNA for this. So we have our standard PGTai offering, which is our default offering. And whilst I'm not going into all the technical details of how PGTai 2 works today, we have several webinars about this on our website and on YouTube already. The main point I'm going to draw your attention to is we do whole genome identification of SNPs. So we look across the entire genome of the embryos to find these individual unique markers, the SNPs. So by having this standard test, we can unlock the extra power of PGTai testing by having the DNA samples, and it's just a simple cheek brush swab from mum and another cheek brush swab from dad. And there's various things it can unlock, but the main one that we're talking about today is this genetic PN checking. Does my embryo have just one copy of mum and one copy of dad in my embryo, and it's a true diploid embryo suitable for transfer? So last week we also discussed the origin of meiotic aneuploidy can also be tested at a chromosome by chromosome basis, and I would strongly recommend, if you're interested, to have a look at that webinar as well. But how effective is it, and does it add value to the whole IVF process? So there have been various studies, and I've just picked out 4 for today, and this is the first one. This was a pure morphological transfer outcome. So as I stated several slides ago, we know that by a visual check, then there is a significant number, and in this study they estimated 22% of IVF embryos and 25% of ICSI embryos were actually visually scored as simply being 1pn. So getting on for a quarter of the embryos in this study created would just be scored as 1pn and then considered not suitable for transfer. So under a fully patient consented study, they took 33 of these 1pn derived embryos and transferred as single embryo transfers. And what they found from these 33 embryos was 9 healthy live births. So this worked out to be basically the same live birth rate as they were having from their visually checked TPNs. Now, just bear in mind, this is morphology only. So they have taken these embryos that were scored as 1pn, but they have gone on to form full blastocysts and healthy looking blastocysts. So this argument is that at least some of the true 1pns would have failed to develop into the blastocyst stage. But their conclusion was that if there are no TPNs available, so still transfer the TPNs first, but if there are 1pNs which reach blastocyst as expected, then they should at least be considered for transfer. And I think, this shows that at least in some cases, 1pNs, or at least what we believe to be 1pNs, must actually be 2pN embryos. More recently, and in fact very recently, there was a massive study that came out, and they looked at literally, 13,000 of 2pN embryos transferred from a vitrified warm blastocyst transfer. But again, this is looking purely at morphology. So they also transferred 435 0pNs and 281 1pNs. Now, their pregnancy rate was very similar across all 3. However, they did notice a marked increase in the spontaneous abortion rate for the 1pNs. And that was particularly noticeable in the late spontaneous abortion rate, so plus 12 weeks. But ultimately, the 0pNs and the 2pNs had identical live birth rates and birth rates. So their argument and their observation was that actually 0pN and 2pNs actually had very similar pregnancy rates and live birth rates after a vitrified warm blastocyst transfer. So again, all of these embryos have gone on to form blastocyst. However, they did notice with the 1PN that there was this higher simultaneous abortion rate. And this was really what was leading to the lower live birth rate, because the implantation rate, the pregnancy rate was the same. They did note, however, that the 0PN group had the higher birth weights and a greater proportion of very large for gestational age newborns. So basically, so again, if there are 2PNs available, transfer those first, but don't discard the 0PNs particularly, and also the 1PNs. Just be aware of the risks of that. So as I say, these were all morphological studies, but we know from other work that one of the leading drivers of spontaneous abortion is actually aneuploidy. So we don't know of these embryos that were transferred which were euploid and which were aneuploid. So this was a study that came out in 2017, and they took some, excuse me, sorry. They took some embryos that had been scored as either 1PN, 2PN or 3PN, and took them through molecular PN checking, looking at, again, the SNPs and having mum and dad taken through to show whether it was true bi-parental inheritance. And what they found is the vast majority of these were actually, out of these 27 that they tested, actually 21 would have been considered at the whole genome level 2PN. So this is a nearly 80% of those which were scored as 1PN or 2.1 were potentially 2PN. And due to the technology that they were running, they could actually then delineate between the whole genome 2PNs versus a chromosome by chromosome breakdown. And they found that of the 2PNs, about 30% were actually true euploids, and of those, 3 or getting on for 40% went on to form a live birth. So these were embryos that may not have been transferred on their PN alone. But they found, and I think this is the really key part to this paper in many ways, is that in 3 cycles, if they hadn't have done this testing and just not transferred on the basis of PN, 3 cycles wouldn't have had a transfer, because it was all of these embryos, they only produced abnormal fertilization events. So you could say that's quite rare, 1 in 200 cycles, but nonetheless, it is significant that there are cycles there which would otherwise not have a transfer, which now would. So they were certainly of the opinion that we can do more to help patients understand the genetic makeup of their embryos. And perhaps most staggeringly, in many ways, is this study, which was PGTM, which is why I mentioned right at the beginning the importance to think about this, not just in terms of PGTA, but any cycle which is creating embryos. They still go through the same scoring for 1PN, 2PN, 3PN, and decisions made on the basis of that. So this is PGTM case where not only are they struggling to potentially have normal fertilization events, they're also trying to find embryos which aren't carrying disease. So we need to find all the embryos we can to maximize the transfer rate. So they took 216 good quality embryos. They took them through a process called carrier mapping, which is a SNP-based test looking for haploblocks. Again, it's a test that we run at Cooper for PGTM. And out of their 216 good quality embryos, 49 of them had initially been scored as 0PNs, 15 1PNs, and 152 2PNs. And what they found, and this kind of backs up some of the work by the Chinese study looking at transfer of 0PNs and their success rate, is that 75% of the 0PNs were actually diploid biparental, and even 42% of the 1PNs were diploid biparental. So a lot of these embryos were actually biparental. And by being able to transfer these embryos, they were able to increase the number of cycles reaching the embryo transfer by 81% and the live birth rate by 75%. Staggering numbers, and yes, it's a small study. But again, we're just showing that by ensuring all the embryos are taken through and given a chance, we should be able to increase our pregnancy rate and live birth rate per IVF cycle. So really, in summary, and to allow enough time for a proper discussion, the summary really from the work that's been carried out to date is it can be estimated, depending on the study and how good your visualization of fertilization is, but up to 30% of visually scored abnormally fertilized embryos are in fact diparental 2PN embryos. So if we take that and we say, okay, that's great, then obviously as in patient management, we do not want to cause known adverse events. So obviously, as I said right at the beginning, there are risks. If it's truly 1PN or truly 3PN, there are risks of an adverse outcome. But if we can check those embryos molecularly, then we have the potential to be certain that we're transferring a TPN, not increasing the risk to the patient, and at the same time, increase the number of embryos available for transfer. And the good news is that with the advancing in technology, this is getting ever easier to do. And so for us at Keeper now, all we need is simply a cheap brush swab from mum and dad, an embryo biopsy, and we can tell you whether or not that embryo is truly 2PN or not. And so with that, I'm going to end my presentation and hand it back to Tony, who will take us to the next next point of the webinar.
Thank you very much, David. Excellent introduction to the subject area there. Really interesting. So I would like, first of all, a little bit of housekeeping. We have our poll that we've been running, so I'll give you some of the results from that, but also we have our first questions coming through in the Q&A. So if you have a question that's based on David's presentation, or if you have a question that comes up in our conversation with Christian and Balsam and Colleen, please don't hesitate to put that into the chat box and we'll try and get to it as we go through. So first of all, the poll, actually. So we had quite a good number of responses for it. And we had, how satisfied are you with your current PN assessment? And it was multiple choice, you could answer multiply. So one person asked, what's PN checking? So maybe you're not on the right webinar for that person. But for the rest of us, we had some good answers. I'm happy with the accuracy of our methods. 14% was happy with their current methods. So not a lot of people happy with their current methods. And then the majority of answers were, sometimes it's difficult to be sure of PN status with visual means alone, including time-lapse. Half the recipients said that. Could be improved with genetic analysis of some PN failed embryos was our highest, but that might be due to the fact that you're here because of the title talk, but still interesting, 62%. Adding PN checking would allow me to consider 0 PN and 1 PN embryos for transfer, which was 40%. So even though people thought that genetic analysis could help, there was a little bit less willingness to transfer just on genetic basis alone, which is pretty interesting. So thanks very much, and we'll move on. And first of all, I'm going to go to Balsam. And Balsam, welcome to the webinar. Great to have you here. And I believe, Balsam, that you had an interesting case that you're going to start off by telling us about. And cases are always great to see because they're quite sort of personal and take some of this science and make it down to real people. Balsam.
Yeah, thank you, Tony, for inviting me in this webinar. Before I start my case, I would like to say one of the main limiting steps in IVF is the availability of viable embryos to be used for transfer in each cycle. And this particularly relevant for patients with poor prognosis due to advanced maternal age or showing poor prognosis to control ovarian stimulation. In these couple, the limited number of oocytes we have in the lab resulted in a significant reduction in their outcome for achieving live birth. And usually embryos bearing abnormal ploidy configuration usually originated either digenic or dyspermic conception or paraffinogenic activation, showing an abnormal number of PNs at the fertilization check. And accordingly, I believe practice worldwide, these kind of fertilized oocyte, unfortunately, routinely discarded. And I believe that generally losses about 10% of the zygote obtained. Despite that, we have PN check that it's been used for a long time since IVF started. So PN's checking, unfortunately, is not a reliable platform to be used. So the case that I would like to present today it was a PGTM case who had obtained 20 embryos from 2 IVF cycles. And all those 20 embryos, they showed normal PN's appearance at fertilization check. 10 blastocysts had been biopsied between day 5 and day 6, tested through PGTai2 and linkage analysis SNP array and direct mutation test since it's PGTM. Surprisingly, in this test, and I'll share the video in a second, 40% of her embryos were digenic. So in another word, they have extra set of maternal chromosome. So if she was a case doing normal PGT for aneuploidy testing, so 40% of her embryos might be reported as normal if they don't carry any additional or segmental that can be reported as high or low level mosaic. So they will be as normal. And then that patient come for transfer and then miscarriage, and then we wonder why. So I would share this with you. If you can see my screen. I'm not sure if you are able to see me or no.
Yes, we can see that. Yep.
Yes.
I can't tell you what it is other than it's an embryo because I'm not an embryologist, but
yes,
you can
I believe
talk us through it.
Can see it. So if I run it from the beginning, so you see nicely 2 PNs appearing, and we could argue this is fragmented polar body, and then nice development, nice division, and then end up with beautiful blastocysts. You're going to see it in a second. No standard IVF, we will transfer this embryo, but having knowing that embryo is abnormal, unfortunately, is triploidy. And this another embryo I would like to share with you. Again, the polar body was not confirmed in this case. We thought it's fragmented or not extruded, but again, we can't disregard this embryo because it's developing very nicely. And again, it was triploidy, unfortunately. So this platform, the PGTai 2+, I believe it's a real add to IVF practice because it will allow us to identify and increase the number of blastocysts, and we can work with patients.
So Balsam, you know, I'm not an embryologist, so this might sound like a naive question. So, you know, you could see the pronuclei quite nicely in those time-lapse images, but you couldn't see the polar bodies very easily. And yeah, and you know, this comes back to actually when we used to do a lot of polar body PGT back in the early days of doing 24 chromosome PGT. And how much reliance do you take in PN checking using the pronuclei, and how much do you like to see the polar bodies at the same time? Is it essential to see all of them, or is it really the pronuclei that are the key?
Well, ideally we would like to see both, but sometimes we rely on the PNs, and we believe it could be if we see like fragmented polar body, or if we can't confirm the problem, we can't consider this as abnormal fertilization, because routinely we check the PNs. I think it's useful to check the polar bodies, but they can degrade it during fertilization process. So PNs is more reliable tool in IVF checking.
Yeah, that was always one of the problems with polar bodies, whether you take them sequentially or simultaneously. And if you took them simultaneously, the first polar body was degrading. Yeah, interesting. So we have a question, an actual question came from Colleen, and it's to Balsam and Christian, and it's your standard approach to PN checking, especially if you get a large cohort with a lot of failed PN checks. What's your approach then? Do you then consider doing anything different with those failed PN check embryos? You know, in the situation where you get a really abnormal number of failures to PN. Balsam and then Christian.
Right. So we usually, when we come, it depends whether it's IVF fertilization or ICSI fertilization. So in IVF, when we have failed fertilization, first we check the sperm binding, we check the sperm survival, we check all the egg morphology, and there's polar body faint PNs. And then the privilege that we have in London Women's Clinic, we have time-lapse incubators. So we culture all patients in time-lapse. So we culture everything, and we observe that if there is any change, if there's late appearance in PNs, because studies shows that PNs could disappear as early of 6 hours after sperm injection, or late of 30 hours they can appear. So we culture them till next day, and then we see if there's any activation. If it's confirmed failed fertilization, unfortunately, we can't do anything. ICSI could be more, we can more observe embryos, because we culture them in time lapse after injection, and then we observe if there's any sperm activation, and if there's any polar bodies exclusion, anything, and then we leave them for 24 hours, and then we confirm whether we have fertilization or no.
Cool, thanks very much. Christian.
I'm going to sort of follow on from that. So I've seen in the question and answers sort of a question from Emma Whitney regarding sort of just growing all embryos to the blastocyst stage. I think that if there's any ambiguity with regards to fertilization, my preference is to simply grow everything to the blastocyst stage and see what makes it. For me, this technology that Cooper's is proposing isn't new. I've always been a fan, those that know me, of SNPs. I've been working with them for many, many years. And ultimately, I think that doing molecular fertilization checks are the way of the future, simply growing everything to the blastocyst stage and doing retrospective checks at that point in time. If we had a conversation previously to this as a group, and I sort of brought up the point that if PN checking was a validated test in let's say a genetic testing laboratory, it would not pass QC. The specificity of the test is so low that you would never run it as a PGS test or a PGD test, for instance. So relying on that at any point in time, whether you have a really low fertilization rate and embryos develop, even if you have a good fertilization rate, and trusting that actually the embryos, like Balsam demonstrated there, SUPN embryos are not always diploid. They are quite often triploid. Tony will tell you exactly what percentage he sees, but it's certainly not 0. 3%. There you go. It's certainly not 0. So I think that essentially what we're seeing with these newer technologies that are coming out is that it's far more robust, in fact, to do fertilization checking on blastocysts, especially if you're doing PGS. And we should be erring away from doing classic pronucleate checks for obtaining embryos. I think it's an outdated way of doing things. And just because we did it in the 1980s doesn't mean that we should be doing it these days if there are better technologies available.
Yeah, I mean, it's always difficult when something comes along that's slightly different and challenges the norm. You know, you need a lot of data to accept that. And, you know, it's one of the things that was interesting about that poll was we asked people whether they thought their PN checking was fit for purpose in maybe not the time lapse, but in general sense. And, you know, that was one of the highest answers to say it wasn't fit for purpose. But at the same time, when we said, if we gave you a genetic test that said that those 0 PN and 1 PN embryos were bi-parental diploid, then there was, you know, half the answers, half the survey respondents said that they would then transfer an embryo based upon that. So there's clearly, you know, some concern that the technology has to be proven, you know, further down the line. You know, if you had a scenario where you had a cycle where you'd grown some PN fails to blastocyst and they were the only ones that were euploid, biparental euploid, would you consider transferring them, Christian Baltham?
100%, all the time. I mean, let me qualify that. I think there needs to be a distinction between sort of a classic PGTai quantitative method euploid and a SNP-based technology euploid. I think that a lot of people, and I've come across people before thinking, you know, I can biopsy a 1 PN, and if it's normal, then I can transfer it. That's actually not the case unless you're doing unless you're using the SNP-based technology to look for correct fertilization. But ultimately, I think that it would be probably, and I think the evidence supports this. I mean, looking at all the papers that David ran through earlier, the one that I like the best is actually Antonio's paper. Obviously, I'm really quite familiar with that one there. I think it's been demonstrated really, really well by lots of groups now that it is a technology using SNPs to determine correct fertilization, to identify diploidy, I think is the right way of saying it, in blastocysts, is absolutely robust and is a better technology than, by far a better technology than PN checking. So I would even go so far as saying that if I had a more advanced, better quality blastocyst that was a 0 PN, that had this technology done on it, I would transfer it over a 2 PN that had the same technology.
So for us, we are hesitant to transfer 0 PN or 2.1 PNs without checking them because we never know whether they are haploid, whether they could lead to molar pregnancy or choriocarcinoma. So it's sort of hesitant in our lab to transfer those embryos. We culture them for diagnostic purposes. We always prefer to transfer 2 PNs, but having this technique, I think it's a real add for IVF patient, that we are 100% we transferring diploid blastocysts. It will give us more confidence of what we are transferring, and then we can disregard the PN checks.
Yep. We had a really interesting question from Emma Whitney, and Emma asked a good question. So if you're taking DNA from couples, and one of the things that I don't think we mentioned quite clearly was that in order to be able to do this PN checking, you need a cheek brush swab from the couples. She says this technology is basically the same as carrier mapping. Christian's favorite subject. It is exactly the same as carrier mapping, and absolutely right. And actually, should we be considering, you know, the same process of taking 0 and 1 PN fail check embryos that grow to blastocysts for PGTM? And I think it's the same question.
You do it already. You do report for carrier mapping. I'm sure you've done it for samples I've had before, where you sort of called
The
triploids.
case
and
that I just
triploids.
presented was the karyomapping, it's PGTM case. So it detected the triploidy.
Yeah, and it would detect haploids in just the same way. So yeah, I think the thing is that, you know, it's taking the technology and taking it to a price point and a sort of single test, which takes it from the sort of complexity of PGTM down to PGTai. I think that's the key point. And, you know, but Emma is completely right. You know, if somebody isn't taking their 0PN, 1PN fail embryos to blastocyst in PGTM, they should be, because the carrier mapping technology, you know, I think we've done more than 10,000 PGTM cycles with carrier mapping. You know, we're fairly confident that it's robust by now. So yeah, I think, yeah, your example is pretty good. And we also had another question, which was, what is the advantage of doing over PCR based check for Ploidy in addition to NGS? The advantage here is that it's one test. You don't have to do two tests. It's the sequencing takes the SNPs and the NGS at the same time. You just have to get the cheek brush swabs. And just in general, what is your actual experience of how patients expect in terms of numbers of embryos to fertilize and the news that embryos have failed PN checking?
Right. So we usually, when we call the patient for fertilization check, we go through all embryology, and unfortunately we explain that we miss the first, especially for IVF. We ICSI, we don't miss PNs if they appear, but with IVF we often do miss sometimes faint fertilization or 1 PN or 2 polar bodies. So we and 0 PN. So we usually advise the patient, again, we can't use it for transfer, unfortunately. So unless the patient has, for example, 14 embryos fertilized, 14 embryos, and then 1 or 2 embryos, they will maybe willing to sacrifice them for, because we missed the PN check. But for the patient who have small number of embryos, it's put us in very difficult situation. IVF, I would be more reluctant because we don't know from where the sperm entered to the egg for fertilization. It could enter from the position near to the spindle, and that's why the nuclear envelope for 2 maternal and paternal, they didn't have the time to develop, so that's why they came on in 1 PN. But for ICSIs, we are sort of hesitant to how to deal with those embryos. So usually we don't use them for transfer, unfortunately, unless we subject them for the genetic testing.
Christian, did you want to add anything else there?
Sorry, I sort of drifted there. I can't remember what the question was. Apologies.
It was what patients' expectations were of, you know, failure to fertilize.
To be honest with you, I don't think patients really, I mean, they have an expectation that really all of their eggs are going to fertilize, to be honest with you. I think that that's what they come in hoping for at the very least. You know, obviously expectations are managed, and we explain to them that's not always the case. But I think that, I don't know if you're coming to the point about, I suppose, educating patients on this technology may actually be a little bit trickier than simply going ahead and saying, you know, we need to take some cheek swabs to do fertilization checking. It is quite a complex, you know, for us as embryologists, even molecular biologists among us, the whole idea of understanding, you know, the genetic status of an embryo is fairly intuitive, but for patients, I think it can be quite a tasking concept to grasp. I've certainly had some resistance. In fact, I would say a complaint as well, a formal complaint, because patients didn't really understand the process that we were going through to try and enable them to increase the number of embryos that they had by doing this extra testing for them. So I think there is some learning, certainly from my behalf. I'm sure that in every center, before we can move exclusively over to a technology like this, is to learn how to convey this information to a patient so that it is easily digested because it's very easy to say, you've got 5 embryos when you speak to them about, with their fertilization results. It's far more complicated to say, sorry, excuse me, that we're going to grow all of your embryos through to the blastocyst stage of development, and then we'll tell you how many of them are going to be normally fertilized, and then which ones of those are going to be euploid for embryo transfer. It's just a mindset change, and working out how to do that, I think, is just coming back to your question, managing the expectations of the patients in a different way.
Yeah, it's so much of what we do is managing expectations of patients. And actually, one of the things that I think is interesting, and maybe Colleen, you could join in as well, is what embryologists' kind of understanding of this is, because you don't need to just understand that you now have euploid aneuploid mosaic, which is our new complexity for trophectoderm biopsies. But if you're looking at these, you have to understand that it's copy neutral events. So events that would otherwise, with just a standard copy calling test, would be euploid. You have to look for SNPs, you have to take the parental DNAs, and you have to then see whether you've got an equal contribution of both parents. And do you think that that's going to be an education? Do you think the embryologists will be prepared to do that? Do they see enough benefit, Colleen?
Yeah, I mean, I think it's an education, not just for the embryologists, but the clinicians and everybody that's involved with the patient care. And certainly from the genetics perspective, it's been a steep learning curve for a lot of embryologists over the last few years as we've introduced more and more genetic testing. But I think by and large, any anxieties or concerns that embryologists have in communicating this kind of information to the patient is ultimately overcome by the fact that we know that we've got to do the best that we can to provide the best chance of pregnancy for our patients. And if that means doing PGTai and having to identify euploid embryos, we learn that. If it means learning about bi-parental inheritance and genetic PN testing, we do that. So certainly there'll be anxiety in terms of feeling that we fully understand the concept and being able to explain that to the patient in a way that they will understand. But that's all an education, and something that I think people are definitely prepared to do if it means we have embryos available for the patient that they would have previously been disposed of.
I think, just to add to that, I think it is really, really important that there is a good amount of understanding before a technology like this is uptaken, because there is the danger that you can run away with it without understanding it and then scare people away. On the whole, what you said, Colleen, I think is kind of nice, in the sense that we are trying to do what's best for the patients, and as long as we can get that across. Initially what I've come across, especially talking to the consultants here at the Eve Well, is that if we say to them, for instance, we're going to test this embryo because it's a 1PN, because it's a 3PN, but it's made a nice blastocyst, and this patient hasn't got very many embryos, so on and so forth, introducing it slowly, and explain to them that we might get more embryos to transfer, they're automatically straight away willing to go for it. But I think, again, there is a huge amount of caution and education that needs to be taken, and I think hopefully, Colleen, that you can spread that through what you're doing.
Cool. Balsam, did you have anything else to add to that?
One more thing, it came in my mind. We use lots of donor sperm and donor eggs in our clinic. So will be Cooper one day approach that, for example, for donor sperm, you can amplify sperm DNA and use it instead of the cheek swabs?
So yeah, there's obviously always some limitations to any technology. One of the questions actually came through was, can this be done on day 3 embryo? And we don't do any blastomy or single cell work. It's all day 5, day 6 trophectoderm. And we can, if we have the semen sample, take DNA from that, and then use that in proxy of a cheek brush swab. But that's not something we do routinely yet. And similarly with oocyte donors, you would need either cumulus cells, or you would need the cheek brush swab from the donor, the oocyte donor. So these things are not insurmountable for donors, but it's not really been done yet for donors so far. And one of the
I was going to say, Tony, there's a bit of a precedent, because I've been involved in a number of PGTM cases, ones that have used donor sperm and ones which have used donor eggs as well. And it seemed very complicated, but it was a matter of us making contact with the donor banks, and either, as Tony said, getting that cheek swab from the donor, be them male or female, and also making sure that everything was above board, sort of consent wise, to actually perform that testing. But it's one of those things that, if it becomes more wider spread in a test like this as part of PGTai, then the pathways to doing that would probably become more clear and easier as well.
Thank you.
Yeah, thanks, Colleen. And one of the things that's interesting about PGTai is that it's long been held that it's never going to be successful because it reduces your number of embryos to select. Now, that's the way it works. Clearly, if you implant every embryo, it's going to work just as well as any PGTai, but you have to go through maybe lots of unnecessary cycles. And this is one of the few instances which you might see a positive in that scenario to PGTai, in that you might end up with more embryos to select from, rather than cutting down your number of embryos. And I think, Christian, we've kind of batted backwards and forwards a little bit, approximately how many extra embryos we might get. Do you have any kind of sort of feeling for how many extra embryos you might get if you check the 0 and 1 PNs?
Well, 0 PNs, I think, are a side effect of IVF and the missing of pronuclear formation. Basically, seldom do you see a 0 PN that forms a blastocyst after ICSI, especially when you've got time lapse. But certainly, if you were to include 1 PNs and 2 PN with a micropronucleus, or 2.1 PNs, however you want to call them, I don't think it's going to be a huge amount. Perhaps maybe a couple of percentage points you might increase it. But if you think about that, as well as the the 3% of diploid or 2 PN embryos that are not actually diploid, you've got a swing of probably 5-6%.
Yeah,
almost certainly it will be a significant increase. I would go out on a limb and say, if this was done routinely on every IVF cycle, you'd see an increase in success.
Okay, I think that's a very kind of positive, uplifting way to finish, because as ever, we have had an hour and we could stay here and talk all evening. And I think it's a great conversation. And I would just like to thank Balsam and Christian and Colleen for joining us. And maybe we'll come back to this in the future when we have enough data to really go back and go through and work out exactly the impact it's made. And we really look forward to that in the future. I'd also just like to thank David for his presentation earlier. And I would like to say thank you for attending. Thank you for the questions. And we'll see you at our next webinar. Thank you.